Oh, good evening, folks.
Good evening.
Welcome once again. And this is Kurt Lannis. He'll be giving us our presentation this evening. But before Kurt starts, just some calendar reminders. We've set up Bible and Science to meet on the second and fourth Thursdays of the month, which sounded pretty benign until we looked at the calendar for November. And of course, Thanksgiving, it's always the fourth, right? And then when we looked at December, guess what the fourth Thursday is? December 25th, right? Christmas Day. So on those 2 months, we'll meet only on the second Thursday of the month. So just heads up on that. The church calendar's already been updated to show those things, and that's as it is. In case some of you didn't hear earlier or just got here, there are some drinks in the back. They're left over from last night's council meeting. They're not tremendously cold because they've been here since last night's council meeting, but you are certainly welcome to them. They are refreshing nonetheless. So please help yourselves. And with that, I turn things over to Kurt and tonight's topic. Thank you, Kurt, for being here this evening.
Well, shall we open with prayer then?
Thanks.
The Lord be with you.
And also with you.
Thank you, Lord, for the many blessings we have enjoyed, and also we come here to learn of your magnificent creation, learn what we can learn of that. And we ask that you, with your spirit, guide us and teach us about your magnificent holiness. In Jesus' name we pray.
Amen.
Amen. Thank you, Kurt.
So this is the second session. The first session, we never really got into too much of the steps, okay? And maybe this will be the last session, depending on how many slides I can get through tonight. So to start things off, I thought I would show a little of my astrophotography. So this is the Pinwheel Galaxy. I actually took this image here from here in Albuquerque. All these little stars and stuff that you see, those are actually in our galaxy. Okay, so it's just kind of impressive when I look at it. You can't see any of the stars in the distant galaxy. Another curious thing is, while I was taking this image about 2 years ago, there was a supernova in that galaxy, and I just between one night and another night, you could actually see it. You can't see it in this image because we had troop on and stuff. But anyway, you could see it out in one of the arms, lit up really bright. Wow. It's kind of incredible because it lit up a big area of the galaxy. Yes, sir.
Well, did you say you took that here in Albuquerque?
I did too.
Really?
Uh-huh.
I didn't think you could get that clear of an image here.
Yeah, it's challenging.
How long an exposure is that?
I couldn't say right off, but it's probably somewhere between 30 to 50 hours, something in that range. So, okay, so we'll get started here on the, oops, is the clicker working? I can click manually.
Let's probably this. Let's see. Yeah.
Oh, it's gotta be there. Okay, okay, great. There, it is working. Okay, so we'll do a quick review. We talked all about this last time, but we didn't actually talk about the Stairway to Life. So the Stairway to Life, it's kind of hard to see. It outlines 12 steps, okay, for life to rise spontaneously. And each step is a scientific hurdle, which calls into question this idea of naturalistic abiogenesis. So I said we're gonna focus on 1 to 3. I guess that's needs updating. I think we'll get through a lot more than 1 to 3 on the starting points of life. So the idea of what we will explore today, we're going to try to understand the steps of the Stairway to Life. And I am reading another book now after listening to the talk a couple of weeks ago. There was a discussion about the immaterial genome. I thought that was interesting. I downloaded this book called Plato's Revenge. And it's about the complexity, of the process by which, cells divide and embryos grow. And how it may not be, there may not be enough information in the DNA to actually make that happen. So, even though this maybe sounds complicated in the 12 steps, okay, it's really just kind of scratching the surface. We're really kind of talking about some of the very basics of life. So the 12 steps, we'll just go through them real quickly. First is the question of formation of the building blocks of life, okay? The second one we're going to talk about is chirality, which is a way of saying handedness of the molecules. Then we're going to talk about the water paradox, which is interesting roadblock to the actual creation of life. Then we're going to talk about how the building blocks of life get connected together into more complicated structures in the cell. We're going to talk about, well, how would those things reproduce? Because, you know, the whole idea of evolution is that, you know, you start out with some, some life that starts reproducing, and then you can bring in this idea of the survival of the fittest. So we'll talk about that. And we'll talk about the nucleotide sequences, which is just the DNA. We'll talk about them forming a useful code, okay, because it's basically like a program, okay, and so it has to form a useful program. Then we'll talk about gene regulation. This is kind of a fascinating subject to me, and I'm not a biologist, but there has to be a process for turning on the various genes, because genes produce proteins. There has to be a process for turning those on at the right time. Otherwise, you kind of get a mess. So we're going to talk about repairing, the process of repair, and then selectively permeable membranes. You know, your cells are basically a membrane, and it keeps all the cellular machinery inside and everything else outside. But it has to allow food and oxygen in, and carbon dioxide and other things out of the cell. So then we're going to talk about energy, how the cells generate energy, okay? And then we're going to talk about the independency of DNA, RNA, and the proteins they produce, and how a little bit about that mechanism. And then finally, the last step is coordinated cellular purpose. So this, oh, just got to think about it, I guess.
Yeah.
No, I
Is there an
could,
hourglass there?
I did, but yeah,
First thing about that.
okay, now I got my hourglass.
It shouldn't be an hourglass. Sorry about this, folks.
Okay.
Sometimes a network wants to, that's well.
Oh,
Come on now.
hmm,
Very odd. Oops. I think.
okay, there you go, that was right. It's cool. Okay, so step one is the formation of the building blocks. So life is built out of 20 fundamental organic molecules called amino acids. And if you look at it on the right-hand side is the acid part of it, and on the left-hand side is the amino part of it. So that's how it becomes an amino acid. And in the center, there's what's called a side chain. So all of the amino acids, they're essential for all the biological processes in living organisms. They're just kind of like the fundamental building block. So the little R group there, what kind of molecules are attached down there determine what the amino acid is. So they all have that one form, but then they have a little R group that's attached to them that make them a specific amino acid. So here's a chart for you to memorize. These are all 20 of the amino acids, okay, and all the names for them. Those are the fundamental building blocks. So what do amino acids do? Well, amino acids, they build proteins, okay? And that's what people are made out of, proteins. So, and it's kind of a complicated process. They form these helixes, and then they form these chains where the helixes wrap around, and then they form proteins, okay? And the proteins do certain functions in the cell. So we talk about the requirements for building complex molecules. So the right molecules have to be present and in the right forms, but one point is that they're rare, okay, and they're not very stable. Okay, so as the first thing you have to do is you have to get these molecules, and we'll talk a little bit about some theories about how that might have happened, okay? So elevated temperatures accelerates the degradation of these molecules. And then also extreme pH, if it's too acidic or too alkaline, then that can also degrade these molecules. And some amino acids are easily oxidized, so you can't really have oxygen around when you're building these amino acids. Now the cells, you got your whole cell, all those amino acids are all protected inside the cellular environment. Okay, so that's why you don't have this problem of rusting. So there's aromatic amino acids, and they can be subject to light degradation. So, and high concentrations can result also in degradation of the molecules. So the early oceans were, which is where some people propose that's where life started, were highly acidic, and they were also quite salty. But that's not favorable to the production of these amino acids. So some of you may remember in 1953, I know, then there was a Miller-Urey experiment, okay? And so these guys, they took basically took a flask, and they passed an electric spark through it, and they put some gases. They said, well, these are a primitive atmosphere, and they put these gases, and of course they had like a trap down there, and some water and other stuff in their little experiment, and they made amino acids, okay? So of course, the way the news is, it hasn't changed in that many years, okay? Oh, these guys know how life was created. It was created right here in this experiment. So we should maybe take a little look at that and see really what's going on here. So there's a few criticisms of this experiment that came about later on. They don't publish their criticisms as much as they do the headlines. But so one was, hey, these guys used laboratory grade reagents, highly purified reagents. Where do you find those in your primordial earth? It's not like 99.9% pure, carbon dioxide or whatever. So the second one is they really didn't have the right kind of atmosphere model. Okay. That probably doesn't matter so much because there's all these other barriers to this. They got a limited diversity of molecules that they produced. There was a couple of amino acids. They say, hey, great, look, this is how life began. So they kind of oversimplified the early conditions on the earth, and there's no mechanism for building more complex molecules in this setup. They just got their amino acids. This one got amino acids. So what they didn't say is they also got a bunch of stuff that you would like. Okay, they got formaldehyde and cyanide and all these things that really are not very beneficial to the formation of life. But that didn't change the headline. So Also, they have that trap that I pointed to. That's how they removed the molecules from the environment where they were being created. So I guess you'd have to postulate that that was also present in your early Earth. So there's another thing we're gonna talk about, I believe next, is the chirality problem, okay? Amino acids produce, you can learn all these fancy words. They're racemic mixtures. It's just like a random mixture. Okay, and life, we'll find out, is not made out of random components. So, okay, so the second problem, or the second half of the first step, is you have to have a concentration of these blocks. So they need to be concentrated in one place so that you can have the reactions between the different components. If they're not concentrated, you're not going to have those reactions. So the natural environments basically dilute the molecules, and the author said, well, it's like trying to make a cake batter in a swimming pool. Okay, it doesn't work out very well. So there's some possible mechanisms, okay, when someone brings up these criticisms, of course, they have to figure out some way that can't be settled. And so they've proposed evaporation. Oh, well, maybe these were little ponds, okay, or it deposited on mineral surfaces, or they were in volcanic pools, but each of these also has its own limitations. So evaporation, that's great for making salt flats, okay? So it's not really great for making concentrated amino acids, but so it can't really just explain what you need in order to get to the next step. So, there's certain properties that are required for certain reactions or processes to take place, and in some cases you say, well, no, that has to be like in a little pool, but then another one you say, well, no, that can't really be in water. Okay, it has to be something. So you have all these, I guess, if your life is evolving, it's going to have to be changing places all the time so it can get to the right environment for the next step. Specific minerals and having a proper pH is critical for all of these chemical reactions to take place. Also scaling these things to a real environment is kind of a major challenge. Maybe you can do it in a test tube, or maybe you can show it, in a petri dish. Okay, but how do you show it that it happened in the real world? So we're going to talk about now, I like this. This is chirality. Okay, so amino acids are chiral molecules. There's right-handed and left-handed molecules. Okay, and just like your hands, you can't superimpose them on each other. Okay, so the unique thing is life uses all left-handed molecules. Okay, no right-handed molecules. In fact, right-handed molecules are either not very nutritious or poison. Okay, so all of the proteins, the 20 amino acids you're going to put together in life, all have to be left-handed. Okay, and all of these mixtures and stuff they're talking about, they don't discriminate based on right-handed or left-handed. Okay, and that's essential for the step of creating proteins. So life is left-handed. Okay, they're all what they call the L form, except one, which is glycine, which is what they call achiro. It's neither right-handed or left-handed. So you have a kind of a problem right there when you're assembling these molecules. So deep, as we mentioned, the right-handed molecules can be either useless or toxic. So all the natural processes we talk about, they make equal mixtures of right-handed and left-handed molecules. And so you're going to build these compounds. What is the chances of you getting all 20 of the left-handed ones in your molecules? So
This is disturbing, Kurt. Does this mean that God is left-handed?
it might be,
No.
be a southpaw. Okay, so, then, when you have these random mixtures, you don't make, very good proteins. So proteins are, they have to be folded in a certain way, okay? And in some cases, maybe we'll see it a little later, they're folded in such a way that there actually electron tunneling occurs between two folds of the same protein. And that electron tunneling, when an electron tunnels from one side, so these things have to be precisely very, a few angstroms apart. When the electron tunneling happens, the molecule changes shape. So this is one way that the body makes little motors. Okay, but that all depends on protein folding. So proper folding in 3 dimension requires left-handed molecules. So I don't know why, but that's what I mean. So okay, so throughout the book they say, well, let's just assume that we got through those other steps. Let's just pretend like we're there. Okay, so what's the next stop? Okay, well, this is kind of interesting because so step 3 is the water paradox. Okay, so what's the water paradox? So water is essential for life, right? Okay, but it inhibits the formation of the biological molecules that you need for life. Okay, and you say, well, that's interesting. Okay, and that's what's called the water paradox. So they have the people that are saying, oh, this all happened naturally, they have to come up with a solution to this. So one hypothesis is that, well, maybe it occurred in these hydrothermal vents. Okay, so I don't know how I don't know how that actually works, because we just went through the things that they need to be concentrated, but now they're gonna be around these hydrothermal vents in the bottom of the ocean. But they say that, well, the presence of CO2 coming out of the volcanic vents could actually act as a dehydrating agent and prevent the water molecules from interfering with the building of the other molecules.
Didn't you say heat was bad?
Pardon me?
Didn't you say heat was bad on the previous slide?
Yeah, it's bad for some of them, right? Okay, and yeah, so that's where you get these environments that are kind of contradictory, right? So then another idea, people said, well, maybe they're in these little microscopic water droplets. Well, I guess those wouldn't be underwater near the vent, but they could be in these microscopic water droplets. And so inside the water droplet, you could concentrate enough of these amino acids that you could actually start building the next step of the structure. I can almost pronounce this. The third is this foraminide. Okay, now I read about this, okay. That's what, if anybody's having their head frozen, okay, cryogenically, that's what they use to keep your head from exploding when they freeze it, okay. They pump you full of this stuff. So they say, well, maybe that could be how we build these molecules. We'll build them in that, so. Another idea is, well, maybe they're on mineral surfaces. If it's on a mineral surface, the mineral can bind to the molecules and can prevent the water from reacting with the chemicals. So cyanamide is another one. Well, maybe that's what it was. Okay, so it kind of seems kind of crazy. So now this is kind of interesting. If you think of this sequence of molecules, there's an immense space there. There's a huge number. Well, we'll talk a little bit about it. So we learned that proteins are made from these 20 standard amino acids. Okay, the average protein contains hundreds of amino acids, and the protein is kind of like your minimal kind of functional block in your body. So the number of combinations of these 20 standard amino acids is really a big number. Okay, so if you just said a chain of 60 amino acids, that's kind of would be a small protein. So that has 2 to the 60th possible combinations and arrangements of amino acids in that molecule, okay? And that's, or 10 to the 78, okay, log 2 of 2 to the 60 is possible sequences. So I thought I'd put this in there for interest. Okay, a pinhead has about 10 to the 17th or 10 to the 20th atoms. Okay, the best estimate of the number of atoms in the observable universe is 10 to the 80. That's in the whole universe. So that galaxy and all the other trillion other galaxies there are in the observable universe, about 10 to the 80 hydrogen atoms. Okay, so this is my vision. If every atom in the universe was replaced with a 60 amino acid chain, okay, 100 or so of them would be useful. So still seems a little far-fetched. So the average protein, now don't post any of this stuff on Reddit or anything, because they'll come after you if you dare suggest that this seems like it can't work. So,
What are the odds?
yeah. So the average protein is much longer, meaning that the combinatorial space is even bigger. Okay. So,
You know, Kurt, I've talked to true believers in this naturalistic origin of life scenario. And I point out some of these statistics. And they say, well, it's not zero, is it? You know, there's still a
Ooh.
chance. There's
It's
still a
not.
chance. And I say, well, my goodness sakes alive, you know, it's infinitesimal. And that's just one, you know, let's say 60 amino acid chain. You probably need hundreds of thousands, millions of them to make the simplest life form. But I tell you, I think these people have more faith than your average Christian. Because the stuff they believe is just phenomenally impossible.
I think your first mistake is, as Scott Adams says, facts are the least persuasive. Okay, you try to persuade somebody, you don't want to try to persuade somebody with facts. Because out of the rank from 1 to 10, it's like on the bottom. People aren't persuaded by facts.
I had a professor nearly 50 years ago who said, space between all the atoms that compose your body.
Yeah.
And there's space between all the atoms that compose that wall. So there's a chance statistically that if you ran at that wall, the atoms would miss each other and you'd come out on the other side. So everybody line up and let's see who makes it. Run.
Or you just do it first on the other side. Because of quantum mechanics, you might just appear on the other side and not have to go through. Okay. That's electron tunneling for you. You don't go through, you just appear.
Let's test that theory.
So experimentally, the portion of random amino acid chains that fold and have biological function is extremely small. So even though you can make these, there's only a small number of them that are useful. So there's kind of a challenge in this protein folding, and if we get through it, there's a little video, so we can look and see how some of them are folded and how they function. But the challenge isn't about finding the one correct sequence out of a fixed number of combinations. The actual problem is that it's infinitely vast number of sequences, and only a small fraction of them will be stable and functional. Okay, so if you look at studies, okay, they suggest that about 1 in 10 to the 11 to 1 in 10 to the 64 might have a simple function like being able to bind a small molecule. Well, we'll see a little later that the functions of proteins in the body are much more complicated than just binding a single molecule. So even if every atom in the universe was used to create, I think I already said that, there'd be far fewer functional proteins than non-functional proteins. But somehow all this happened in this little pond or by this hydrothermal vent.
The head of a pin.
Yeah. So step 4, so now we just assume that steps 1 through 3 actually happened. Okay, we get to the next problem. And that's the linkage of these various building blocks. They have to be linked together, just like DNA is linked together in the chain. So the linkage problem is how do these reactions consistently produce the correctly assembled molecules that you need for life. And so not just occasionally produce them, but it has to be consistent. So the emergence of life depends on things like RNA and DNA and proteins. And random chaotic chemistry doesn't really cut it, and it doesn't help the process. It really hinders the process. So the next part of step 4 in building these RNA or DNA is cross-reactivity. So in your primordial soup, there would be a lot of different mixtures of a lot of different organic molecules. That was the idea. We just make a lot of them, and one of them, you know, beats all the other ones up in the scheme. Okay, so there'd be a lot of different. So the building blocks and proteins would be present alongside all these other things that are going to be like poison, or that might bond to. You might have the right sequence there, and along comes some junk and bonds to it, and now you got junk. So it's a high probability of wrong reactions going on, even bigger probability of wrong reactions going on than the reactions you want. So the second issue is dilution, and we kind of talked about this with the amino acids, but there would be extremely low concentration if this was all taking place in the ocean. Okay, and in order to build these polymers, we need a higher concentration, and that, as we talked about it, evaporation or freezing has been posed that maybe that's how these mechanisms, that's the mechanism for getting the concentrations you need. But the overall dilution issue is still one of the significant issues. So the fourth part of the step 4 is the formation of non-random polymers. Polymers are just connections of these amino acids. So DNA and RNA, they're not random chains. They're highly specific sequences. And what a lot of people mention is that they contain encoded information. Right? It's not just a random sequence. So without, in life, you have these existing genetic systems, okay, that direct the synthesis and the construction and the replication of these, okay? So in your prebiotic conditions, you're probably just going to get a bunch of random junk polymers. But you could always appeal to the multi-universe, infinite number of universes, so you can divide all these big probabilities by infinity, you know, and you can get what you multiply by infinity and get a good number. So the odds of creating specific functional sequences are astronomically low. Kind of the next session, I think this is the last one, is there are thermodynamic barriers to doing this, okay? The body takes care of that because they have enzymes that promote the generation of these things, okay? But thermodynamics governs the feasibility of these things getting organized. So the second law of thermodynamics says order does not arise out of disorder. Okay, disorder arises out of order. Okay, that's how you define the direction of time. Okay, so everything results in more randomness as the process goes on. And some people say, well, you know, there's billions of years. Well, all that does is make the problem worse. Okay, you have a lot more time for it to go wrong. Okay, so there's a problem with thinking about, well, our life is based on DNA, and there's a problem with that. There's no pathway, you know, from DNA, okay, to to life, okay? DNA is the code, okay, but it doesn't, it needs RNA to generate. And also DNA is not stable in of itself, okay, and it needs, we'll talk about this, it needs all these repair mechanisms, okay, to keep the DNA together. So some people have proposed, well, maybe life didn't start with DNA, maybe it started with RNA. So RNA is the molecule in your body that copies the genetic code and takes it out into the cell, and it manufactures proteins from a copy of the DNA. So the RNA makes a copy, takes it out there, builds a protein. The DNA doesn't build a protein itself. So they said, well, maybe it started with RNA, and that RNA molecules could have been the start of life. Well, that's interesting because there's no way backwards. Okay, so maybe they had their RNA world. Well, how did they get to their DNA world? Okay, there's not a path for that. So, but anyway, RNA polymers they're unstable in water in the prebiotic tims, and they're prone to hydrolysis. That's where the water molecules attach to places you don't want them in your RNA molecule. So the second law of thermodynamics says that the system is going to become more and more chaotic, not more and more ordered. So, so there's a peer-reviewed article by this Dr. James Tour that mathematically demonstrates the limitations for these creation of proteins. And he said, even if you had one formed, it would degrade in a few days, okay? So it's too quickly to serve as a stable environment for creating more and more proteins. So all of these polymers, polymers is a fancy name for a bunch of molecules connected together, have this problem, okay? That energy is required to counteract decay of these molecules. And it takes a lot of energy to counteract the decay. So then there's, now we're to step 5, we just assume 1 through 4 all work. We're on step 5, okay? This is how do these things reproduce? Well, this is kind of the chicken and the egg paradox. So life relies on these RNA and DNA for reproduction, and the cell uses intricate molecular machines, and we'll see a little bit a little later about how some of those machines work, okay? Most machines are proteins, okay? And they're encoded from DNA. Okay, that's how they get manufactured. But if you don't have DNA, okay, you can't get the protein, and if you don't have the protein, you can't get the DNA. So they have to all be there at the same time. So there's a circular dependency. So of course, the prebiotic environment doesn't have any of this stuff, okay? So it seems that this idea of reproduction so that we can have selection of the best and fittest molecules is kind of maybe impossible. So we talked a little bit about this, the RNA world hypothesis, that those were the original self-replicating molecules, and those were the foundation for early life. But you can't get there until you get through all the other steps. So let's talk a little bit about the RNA world hypothesis. So authors put it, this is the worst theory of evolution of life, except for all the other theories. Okay.
Just come from DC, so I know, but.
It's too complex a molecule just to have occurred randomly, okay? It's inherently unstable, okay? We need long sequences in order to get to reproducible molecules. You have to be pretty complicated molecules, okay? The best they've ever been able to do on generating RNA that can reproduce is 95, it's 190 long, and it's able to reproduce a 95 nucleotide stretch. Well, we already looked at that in terms of, 60 amino acid molecules. And now we say, well, the best one that we could find that could reproduce is 95. So that's, 2 to the 35 more. So it's far too long to have arisen through any conceivable process of random assembly. Something had to guide it, I guess. So step 6 is now you have these sequences. We got through all steps 5. Okay, now we have these sequences. They have to produce some kind of, useful code. Okay, these can't be random. They have to be useful. So it needs a genetic code decoding mechanism is required, and the replication machinery must also be present simultaneously. So this is where the authors say they coined the term that this is irreducibly complex. Okay, that the minimum complexity that you need, okay, for this process all has to occur together, and we'll kind of see that in the video on ATP synthesis. So ATP is the energy that your cells use. It generates this molecule called ATP, and then it oxidizes it and turns it into another molecule, and then the cell rebuilds that molecule. So there's a whole mechanism in the cell for creating the molecule, ATP molecule, so that other molecules can use it. Well, if you're thinking about evolution, why would you build a mechanism, which is a pretty complicated mechanism, we'll see, that makes ATP when you don't have anybody to use it? Okay. And if you have somebody that needs it, well, then you better have the mechanism that makes it. So, this whole idea of how this code could emerge really needs some further explanation from the evolutionists. So, and theories like a frozen accident, they have this frozen accident theory. Oh, well, once you created that molecule, then it would magically preserve itself, and it would just keep that way. It would be reproduced. Okay, and you think, well, that seems rather unlikely. So, step 7 here is a means of gene regulation, as I mentioned before. So a gene, okay, you need a way. A gene is a sequence of DNA, okay, that encodes for a specific protein, and you need a means to turn that on and turn it off, because you don't want that protein being generated all the time. You only want it when you need it. And so, that process in life is done by gene regulation. And there was many years ago, they were saying, the DNA, only like 10 or 20% of the DNA codes for proteins, and only 10 or 20% of the DNA is useful. And of course, the evolutionary guys would say, oh yeah, that's what you would expect. 80% of your genome is junk, and it's just leftovers from evolution, okay? And that's what we would expect, okay? Well, it turns out, then they found out later, that the 80% of the genome is involved in regulating the 20%, okay? And it's not junk at all. In fact, it's quite essential. So gene expression, of course, we've talked about this, is the process by which the information in the gene is turned on to produce a product, either an RNA molecule or a protein. So In single cell systems, uncontrolled gene expression would be not only wasteful, but it would be potentially detrimental. So assuming we get there, okay, we need a means of reproduction and repairing biopolymers, okay? We need an error correction scheme. And we're gonna get to a little video here that describes that. So that's probably the most glaring omission in these prebiotic systems. Well, how does the error correction arise? Because it's necessary to maintain the integrity of all these molecules. So modern life uses enzymes and they have proofreading to ensure the fidelity and replication. And error rates are as low as 1 in 10 to the 9, okay? In an RNA world, okay, the error rates would be like 1 in 10 to the 3, okay? So as soon as you created a molecule, it would get destroyed. So you have this error catastrophe where the sequences are lost faster than they're created. So without correction, even RNA in their RNA rule would devolve into useless variants. So it's the whole idea, the whole thermodynamic idea of stability versus activity is kind of ignored in the RNA world. So now we get to watch a video. This is
Well, I
kind of,
pray this works.
hopefully it works. Let me see, do I have to click it?
I think you do.
Maybe I should turn on the closed captions.
There we go.
I'll turn it on.
We're working.
Oops, no, I won't. No.
Every day, we are bombarded by environmental agents that damage our cells. Things like UV radiation and chemical exposure, such as air pollution and cigarette smoke. Even the byproducts of metabolism, such as free radicals, can damage cells. These damaging agents can make changes to the DNA in our cells. DNA encodes the information needed to make proteins, which are the building blocks of our body. To keep 2 meters of DNA organized within the nucleus, it is wound around special structures called histones, much like thread on a spool. Damage to DNA can include single-strand breaks, double-strand breaks, changes to the DNA code, DNA kinks, and DNA sticking together. It has been estimated that an individual cell can suffer up to 1 million DNA changes per day. A special sentinel protein called p53 is recruited rapidly in response to DNA damage. p53 slides along the DNA until it finds a critical site. It then binds to this site. p53 then sends the message to halt cell division until DNA is repaired, or if damage is too severe, the cell is destroyed. p53 is therefore known as the guardian of the genome. If the DNA damage is able to be repaired, a very complex and highly regulated sequence of events follows, which is orchestrated by 3 main molecular machines. This is machine number 1, which is searching for a broken DNA end.
They actually make those noises.
Once it has found the broken end, machine 1 activates this molecule. Once active, the molecule participates in a series of interactions leading to machine number 2. Here it is again, this time activating another molecule. This is machine 2, assembling into its active form. Machine 2 builds a chain to link to machine 3. Here is machine 3, which has also built a chain. The final molecule in this repair complex, BRCA1, forms from 2 identical halves, which come together and bind. BRCA1 then begins the recruitment of further complexes that carry out the actual DNA repair. After a long chain of events not shown here, the DNA is finally repaired.
So that mechanism, of course, is created by the DNA, right? The cell creates all those mechanisms, but without those mechanisms, the cell can't survive. And now they kind of like one of the questions on is how did these original cells get built? Okay, and this sounds like, you know, well, maybe they're built on these micrometeorites, okay? Yeah, so micrometeorites come to the earth and like form dust, and they said, they've observed that if you put some fat molecules on these micrometeorites, they make these little corpuscles, okay, and those kind of look like cellular membranes. They're missing a lot of the functions that are in cellular membrane, but they said, well, maybe that's how the original cellular membrane, of course, then it also had to encapsulate all the right stuff inside that cellular membrane. And then another idea that comes up is this same kind of recycled idea of little tiny drops of rain. Okay, they look like microscopic droplets, and they could capture the cell, and therefore it could develop that way. So, but all this overlooks how errors in the RNA would propagate unchecked without error correction. So errors pretty much dissolve any selective advantage you get in the cell. So they treat the assembly as a passive process, but sustaining it requires all of these things operating together. So in summary, while the headlines amplify these little minor lab, oh, we did this thing in the lab, and you see them, I saw one the other day. They said, oh, look, we solved the origin of life. This is how it occurred. They're rather simplistic, okay? And they perpetuate the hype by really sidelining any of these core issues that are involved in the creation. So the James Tour argues that the origin of life, that they've seen billions of dollars of investment over 70 years trying to solve this problem, but they still don't have a grounded proposal for how this all occurs. And the thermodynamic problems and the error correction problems just make things implausible. So these issues really need to be addressed if they're going to try to develop a naturalistic view of how life was created. So the next thing we're going to talk about is the
Kurt,
Yeah.
I was wondering, would it be possible for 7:23? Could you hold off on that and we could take questions?
Sure, if you like.
Would that be okay?
If anybody has some questions, yeah.
Does it go till 8 like last week? Or does it go till 8 or does it go till 7:30?
Goes till 8, right? Do we go till 8 or go to 7:30?
8.
Oh, we go to 8?
Yeah. We have in the past.
Oh.
No. I'm afraid I might finish.
You're hoping to.
You know, when I started it out, when I did that first section, I thought, oh man, this is gonna take 5 classes, you know?
Well, if I can ask a question.
Sure,
When you were talking about the repair mechanism,
uh-huh,
it occurs to me that maybe that introduces yet another chicken and egg conundrum. Because unless you have those repair mechanisms, the rest of the structure isn't going to survive. So it doesn't,
right,
which one has to come first in order for the whole?
right, they both have to come together.
Yeah, what are the
Yeah.
odds? Right?
Well, on top of all the other astronomical odds, yeah.
Precisely.
It seems, what do they call it in science fiction? The suspension of disbelief.
Yeah. Yeah. Sometimes in politics, so.
You know,
How does it know it's in air? I mean, you're producing something,
oh,
what's your standard that you're measuring it against? Against
Oh,
something that
yeah,
doesn't exist?
well, you know, the air guys die off and the strong survive.
Maybe they guess right.
You know, we're dealing with a theory from the late 1800s, right?
Well, and this explains how to, you know, times many orders of magnitude, how you get one cell. How do you expand that to an entire organism,
Right.
say a squirrel or a
Yeah,
human?
there's a lot, it's just complexity on top of complexity. Like I said, even however complicated this stuff sounds, this is really the very basics. As testified that an engineer could kind of understand it.
Oh,
Right.
picking on the engineers.
Okay,
You know, what's so sad is that, you know, the Miller-Urey experiment took place at what, 57,
53,
53?
yeah.
And you know, if you read in a biology textbook, in all of the 50 states, so one adopted here in New Mexico, you could go down to the book depository here in Albuquerque and look at the textbooks. And when they address the Miller-Urey experiment, they might mention that the early Earth conditions were maybe not as depicted in their original experiment. They might draw attention to what's called experimenter interference with a drain cock. And that's about it. And I remember, and I don't think I'm alone in that, when I first read about that, maybe just some minds just kind of extrapolate that. Oh my, we got a couple amino acids made here. Well, surely it's just a matter of a little bit more experimentation. You get more amino acids. And at that time in high school, in heaven's sakes alive, we didn't cover any of this stuff that you mentioned because it wasn't widely known. But my point is, I think it allows young people who don't have recourse to this type of information to just have the assumption that no, these naturalistic processes can explain not only the origin of life, is still, you know, light years from a living mammal, let's say, but they, in their brain, some of them say, we don't need God. We don't need an intelligent designer. All of this science, which doesn't use any basis for faith, can't explain it. And I think that's a real tragedy. So anyhow, most of us here, we probably all have grown children, but I think it's maybe falls upon us to help our grown kids explain some of this to their children and our grandchildren, because they're not getting it in the public schools. And I think it's a real tragedy of what's supposed to be
Well, it's
the
kind of,
purpose of education.
you gotta look at it as kind of humor. You know, this is Charles Darwin, you know, went to the Galapagos and saw some birds look like some other birds. And
Right.
said, oh, wow,
Uh-huh,
that means we don't eat God.
right.
You know, so, okay, so we talked a little bit about how these cellular membranes might be created on micrometeorites or whatever, but cellular membranes aren't simple little structures.
Huh.
Okay, and they have all these. Not only does the membrane have to keep the outside and the inside, but it has to let what the cell needs in, and it has to dispose of the products of metabolism. So all of that stuff has to go on there. So this is kind of diagram. Whoops, what happened there? We'll go back over here. So it's kind of interesting how the membrane is formed. It's got these molecules that have a hydrophobic part, okay, and a hydrophilic part. So part of it, so they connect together in the middle, the parts that like each other, and the hydrophobic parts are on the outside, okay, which makes it basically waterproof. So let's talk a little bit about it. So selective membranes, this is step 9. It's essential to the origin of life. And they have to be selective. They just can't say, well, I'll keep everything out, okay? Which is what a simple membrane would do. So it contains essential molecules and it has to allow those in, and it has to allow a selective exchange with the environment, okay? So lipids that's what the cell membranes are made out of, fat. Okay, they can form closed vesicles in lab settings, but that's really a crude approximation of what a cellular membrane is. And they require purified concentrated conditions, not quite what you'd find. So they can self-assemble in vitro, okay, but they're not a cellular membrane. So no cell, this is kind of interesting. No cell has ever been observed that creates this functional membrane. Okay, your cellular membrane on every one of your cells was inherited from the parent cell. Okay, and then there's mechanisms to expand it, but there's no mechanism to create it. So you have to say, well, gee, how did these cellular membranes get big? So they're inherited, they're used as a template when the cell divides so that the cell can manufacture more membranes, but they're not manufactured from scratch. There's no process that makes a cellular membrane. So it's kind of like, whoa, I guess if that mechanism evolved, it's disappeared somewhere. So anyway, he says, just as every cell comes from a cell, so does every membrane come from a membrane. Kind of interesting. So where did the first one come from? Now we're going to talk about a little bit about come back to these rainwater. They're one of their favorite things. Well, maybe this happened in little droplets, okay? And that's how the compartmentation, and somehow the rest follows from that, okay? But it fails to account for how those structures withstand the environment and the thermal fluctuations and how they maintain their integrity against all the forces that are trying to disperse them. So, and if you don't account for these, then you just have these speculative models. And whenever something is discovered, somebody says, well, maybe this is the way it happened. So we talked a little bit about this. It says one proposal was that the surfaces of micrometeorites could help assemble life's blocks. So basically the surface has a lower energy than the rest of the or has a higher energy state than the internal molecules in a substance, okay? And that can that surface can affect how materials interact when they touch the surface. So you can have wetting, or you can have droplets form, and so this is kind of the idea. I don't know how it became micrometeorites, and it just couldn't be like regular dust, but but one of the things they've observed, and they said, oh, this is probably how it worked. When you put fats on these surfaces and they make little globules, sometimes they connect them with nanotubes, and they said, oh, well, look, these things could communicate with each other, okay? Except there's really nothing inside of them but a fat membrane, you know, anyway, all they're really talking about is how can we get the molecules together to form the cellular membrane. It ignores the big obstacle, which is again the entropy, the tendency to randomize everything, and making complex structures from chaotic mixtures doesn't really happen. So one thing I read in the book I thought was interesting is they said if you took a whole bunch of living cells and you put them in a blender and you mixed them all up, okay, you would have every ingredient you need for life, and it would all be in the right proportions, okay, but you really wouldn't quite have life. So basically, the tools that the cell needs didn't exist, okay? So it's hard to explain how this happened, actually. And it fails to discuss how, I already repeated that slide there. So entropy favors the disassembly of organized structures, not the assembly of organized structures. So I think I'll just press on and we'll do the last couple steps. Because we have almost a half an hour. So means of harnessing energy. So the process requires three essential components. It requires a membrane to maintain an electrochemical gradient, which is maintained across the cellular membrane. It requires the machines to do the transport to pump protons across the membrane. Okay. ATP synthesis is what we're talking about, is used to convert the stored energy in the gradient into ATP, and then ATP is like food for the cellular processes. And all these things have to work together. So now we get to watch, food is oxidized and the electrons flow through the membrane back to the outer world, okay? And the flow powers, we're going to see the ATP synthesis machine. The powers these proton pumps that concentrate protons on one side of the membrane and make an electrical gradient across the membrane. And it's like a battery, and so they flow back and forth across the membrane. So we're going to look at this video, because videos are fun. And this shows how the cell manufactures this molecule ATP that's used. And what you'll see is it's not just one simple molecule that manufactures. This is a complicated process. And if one part is missing, it doesn't work. Okay. So all the parts had to be created at the same time.
Called the F1, F0 ATP synthase, which uses the proton gradient.
You see, I gotta start at the start.
You drag the red dot to your left.
Yeah, I'm trying to. Why do we want to do that? I want to drag.
Drive the synthesis of ATP molecules.
I don't want to be dragged.
Mm-hmm.
What is that?
These ATP molecules.
Wonders of YouTube.
Huh?
The wonders of YouTube.
Yes.
No, he's having a problem because it's not a Mac.
Molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die.
So this is a cellular membrane here.
Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are 4 protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4. Proton pumping requires energy, and the 4 protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the 4 complexes are collectively referred to as the electron transport chain. Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits 2 high-energy electrons in complex 1, where they are passed along a chain of redox centers.
Clear as mud, huh?
Yeah.
Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal 2 reasons why an electron moves from the top redox center to the bottom. First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center. A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons. The last redox center in complex 1 donates 2 electrons to a coenzyme Q molecule.
Did that cost me?
Complex 2 is similar to complex 1 in 2 important ways. First, high-energy electrons also enter complex 2 via a byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons.
This is gonna be on your final. Pay attention.
Coenzyme Q molecules from complexes 1 and 2 donate their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through 2 redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving 4 electrons converts a molecule of oxygen to 2 molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space.
That's why you breathe oxygen.
In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. In this animation, we have explored each protein complex in isolation, but in reality, they are very densely packed. Together, they effectively make the entire surface of the inner mitochondrial membrane a giant cellular power plant.
So, I guess the.
Or the F1, F0 ATP
Go to
synthase,
the next one.
which uses
Okay, so, I guess the point of that is, all of those things work together, right? Also, those things are inserted into the cell membrane, and there's a whole function that gets them inserted in there. How, when you're building up the membrane, you have special processes that basically build those structures, fold those proteins, and then get them inserted into the membrane. So it seems just a little complicated to have happened actually.
Let me just, if I can, Kurt, put that into my terms. Those 4 structures that were just described would have had to have evolved
Together.
perfectly the first time in
Right.
order for anything to have survived.
Right.
So if there's something a little bit off in these 4, nothing would have ever lived
They would not work. And
on
then,
top of the other.
right, but it's
Nine
producing,
steps.
and also it's producing a molecule that's used somewhere else.
Right.
And why would you have a mechanism produce a molecule if there was no place to use it? So the guy that's consuming it also has to exist. Okay, so all those things all have to exist together, and they can't just be, they just can't evolve that way.
What are the chances of that being accidental?
Right. So,
Kurt, you know what blows my mind, but y'all seen, that's taken decades, 3-4 decades to fully understand that electron transport chain. And you got some really brilliant individuals who have figured all that out, scores of. And what really boggles my mind, and you guys might want to ask yourself, why is it that all these brilliant people could come up with that and not see the necessity of an intelligent creator? I don't fully understand it. At least they could be truthful in the textbooks with their students, that's my big push. At least they could be truthful and say, we don't know how all these things came together. It's a mystery here, a mystery there.
and this is just a simple.
Yes.
Well, maybe it's not simple, but it's just one of many thousands of these things that are going on in your cell. And that little carousel that's going around pumping electrons, they said that's going about 7,000 RPM, poof, in your cell walls. And there's like millions of them in each cell, all pumping. So it's pretty amazing. So, there's all this interdependency. We see it everywhere, okay? The cells rely on a mutual collaboration between the biochemicals to survive. There's mutual dependency among them. The different proteins themselves are transcribed from the DNA. And the machinery necessary to do the transcription and create the RNA and are all, made of proteins that can only be built from the RNA and the DNA itself. So all this stuff it looks like a system. So it requires enzymes, okay, and none of these enzymes would exist in the prebiotic world those decode the instructions and basically build all the processes in the cell. So if you have all the 10 previous steps you've made them all, okay? Now we have genes that code for metabolic functions and repair and regulate. How all of that stuff is embodied in the cell. So genes have feedback loops and regulatory proteins. We talked a little bit about gene expression, and all of those metabolic regulation agents all have to work together. So coordinated cellular purpose marks the leap, from chemistry. Cells have a purpose. It's not just the cell by itself, okay? The cell has a purpose, and they're combined together, to make the organism, okay? And there are techniques by which they are organized and how structures are built by cells that basically cooperate to each other. So each cell kind of has its job in the larger mechanism. So you just keep adding complexity on top of complexity. So they say for the minimum required number of genes for life is about 490 some genes, okay? And then you have all the mechanisms for transcription and all of these, which are also made from proteins. So it's pretty much an elaborate system, and all these things have to kind of work together. So step 12 is the coordinated cellular purpose, which the subsystems act together in a unified kind of functional manner, and they're just not a collection of random cells. And each cell has its own structure and its own characteristics. The cells in your skin are not the same as the cells in your retina. So integrations of functions in modern cells, metabolism, replication, energy conversion, maintenance, all work together in synchronization, and they're all coordinated, and cells communicate with each other, okay, so that they can do this. It's not just the cell doing its work by itself. It communicates through its neighbors through production of hormones and other signaling techniques. So anyway, it's just the coordination, how did this coordination happen in evolution? Okay, it's really not clear how any of this would happen. Anyway, I think that's pretty much sums it up. Okay. Any more questions?
All this stuff that Kurt's gone through, the end game here initially in the 20th century and the first decade or so in the 21st is, okay, we're going to explain the origin of the first cellular life form. So they were thinking about, and you should be thinking that, okay, let's say some of this naturalistic kind of coming together as possible, and we're going to get an amoeba. We're going to get one of those protists that maybe some of you saw in a biology class in a microscope, okay? This one cellular life form. But then you got to start thinking, well, my goodness sakes alive, a worm. How many different types of cell types does it have? And they have muscles, they got nerves. So you go from an invertebrate like a worm to, let's say, a vertebrate like a mouse. Holy mackerel, again, the complexity just cascades down. Because there's no real explanation on how all of these coordinated systems come together to form an invertebrate or a vertebrate. Initially, it was to try and explain, how do we get a protist, a single cell organism? So, like 99% is missing. right?
Well,
Now, people don't know that, they don't realize
there's
this.
also some interesting signs because Darwin suggested that there was this tree of life, right? So you start with simple organisms, okay, and they evolve into more complicated organisms. So you end up with this tree, and you got monkeys over here, you got men over there. And so the idea was that as you would see all these common structures in the root of the tree, and then you would see how those common structures evolve, okay? You'd see, oh, well, you know, a monkey is just like a man, except it has these three changes or whatever, okay? Well, it turns out when people trace the genomes, okay, it's not like that at all. It's more, there isn't like this progression of this tree. Okay, and people that have studied plants and looked at different plants and they look at it and they said it's more like they tried to draw a diagram of how all these things are all related, and it's more like a network. It's more like somebody took pieces of this and used it over here, and they took pieces of that and used it over there. It's not like a simple progression. And there's other things that are kind of show up in the timeline. It's like there's a thing called the Precambrian explosion. Okay, so you know, if you look at the fossil records for many millions of years, you had trilobites, and nothing else. But then about I think it was 200-300 million years ago in the Cambrian area, there was an explosion of all this diversity of life. And so how did that all take place in such a short period of time? And how come there's no history of a slow evolution, which is what you would expect to see?
That's when the internet was invented.
Al Gore.
Yeah, he was Precambrian.
Yeah. Yeah.
That's a question about sequencing. How does God accomplish this? Has any thought been given to, because you can look at it several different ways, one of which is all this was instantly put in one place, or did it happen in steps, sequential steps?
Yeah.
Did it happen in the 12 steps, or we get to this place, there's a problem, God figures that out. Is it like God's Edison experiment station where he spends all this time figuring these things out, or did this model come from somewhere else?
Well,
in various places.
I thought about some of those things, and if you just take a look at the world today, you have all these ecosystems that are all kind of coupled together, right? And so the thought maybe occurred to me that, well, maybe, the process of making a world has to be something like that, right? If you're going to set of laws in place, okay, and you're not going to just magically make everything appear, then maybe there has to be, and maybe there's a natural sequence that things have to be built up so that you have these continuous ecosystems.
Well, the follow-on question is really about, is evolution a piece of God's toolkit? So at some point, does he, after seeding or creating this miracle, allow the miracle to proceed down a path that he knows is going to happen, which is all these changes occur? Because all these changes occur all the time, every day, everywhere. And there are trillions of microbes, viruses, whatever, that are out there changing it. You surely wouldn't, maybe he does, I don't know, want to tinker with
I think
everyone.
that's the theory of like theistic evolution. Did God accomplish his plan through an evolutionary process?
Right.
But we don't really see that. There's an example in the book that I didn't get to where they try to look at how organisms develop immunity, okay, or resistance to drug. Malaria has developed a drug resistance, okay. So these people studied how malaria developed a drug existence, and I think they say the average, malaria infected person has trillions of malaria organisms in, right? And of course, there's millions and millions of people that have malaria, okay? And so there's a huge population of malaria organisms in the world, and there's a lot of generations. So that they've studied how did malaria develop a resistance, and I don't know what the drug resistance it developed, maybe it was quinine or whatever. How did it develop that? And they found that it was a change in 4 genes in the malaria organism, okay? And so they could say, oh, 4 genes were changed, okay, in this organism to give it resistance. And it turns out those 4 genes that were changed actually broke a mechanism, because the drugs they used for malaria interfered with certain processes that were going on in the mitochondria where energy is created in the malaria organism. And so the malaria organism developed a mutation in these 4 genes, okay, that gave it resistance. But it gave it resistance by breaking a mechanism in malaria that is important in bugs' metabolism. So the resistant bugs are a lot less fit than the non-resistant bugs. Okay, and so what they did is they went out and calculated out, well, to change 4 genes, how many generations, how big a population size did you need, and how many generations did you have to bring about the mutation in 4 genes? And so then you can draw a parallel to that. Well, okay, there's a lot more than 4 genes difference between a monkey and a man. Okay, how many monkeys did you have on the planet, or how many years, okay, that natural mutations could turn monkeys into men? Okay, and it's just not conceivable that that happened in what the supposed timeline was 60 million years for the descent of man. I was reading this other thing, it was kind of interesting. They said, everybody believes in this evolution of man to monkey because this artist drew that picture. And I know everybody seen the picture of the monkey. It says, so some artist draws this picture and everybody looks at it and says, oh, okay, that's how it happened. Look. It starts as a monkey, it's more and more, now it's a man. Of course, that makes perfect sense.
Well, somewhere along the line, science and religion have to be compatible. Because God's given us a brain, he intends it for us to use it, we have used it to do all the wonderful things we do.
Right.
So, at one point, do we say, some of that's been set in place and some of that is running and allowing us to see what's going on?
Yeah, no, I believe that God gave us a brain, we ought to use it, So I don't think he would find objection to us using it. This book that I'm reading on the immaterial genome is kind of interesting because it suggests, well, maybe God has a continuing role in life, that is, very, very fundamental. It's in the actual, gestation process, and that because they're arguing that it comes from the coding. Some genes can code. A single gene does not simply make one single protein. They said some genes can make 100,000 different proteins of all different sizes. So the question was, what controls the expression of 100,000 different proteins from this one gene? And they start to make a kind of a calculation based on the complexity, how many different proteins could be built, and the complexity of the machine, whatever the machine is that's driving this process that's expressing the genes, if that machine was as big as the whole universe, it wouldn't have enough states to be able to drive the generation of the organism. So they're saying, well, if it's not coming from inside the cell, if the information is not inside the cell that controls it, where's it coming from? So, anyway,
Hey, Ed, I've noticed that you have brought up that home before. So it's an area of interest in you and maybe a little perplexity that you see. So how about in 3 or 4 weeks, you do a presentation? And you explain it all to us.
I do that
Your
with
research.
I asked one too many questions.
So,
It just occurs to me that maybe the only thing that's evolutionary is human knowledge.
well, yeah,
You think back to my grandparents' generation, the idea of human flight was impossible. Human knowledge hadn't advanced to that point until they entered their adulthood. And now it's as commonplace as it could be. And who knows where human knowledge will advance in the coming generations as we begin to grasp some of these things. But these things aren't themselves evolutionary. It's just our understanding of them is.
in a way, it's kind of like the revelation of God, right?
Huh?
It's through man's inquiry, he's coming to realize, and a lot of people have come to realize that there's something here that doesn't seem like it could happen the way people are saying it could happen. Well, would you like to close for us, Mark?
Sure. Real quick reminder, we'll come back together again, not on the 4th Thursday, but the 5th. Because of an anomaly in this month, so we'll be together on the 30th of October. Not quite Halloween, but close enough. The Lord be with you. Let us pray. Lord God, we thank you for what you have created. It's beyond our comprehension in its totality, and the more that we learn about it, the more that we explore what you have done, the more marvelous your work becomes in our eyes and in our hearts. So Lord God, help us to go out into the world and share this wonder with the others that we meet, our brothers and sisters of this world, and help us always to hold that sense of wonder so that we look for more and more evidence of your divine excellence in all that exists around us and even within us. In Jesus' name we pray. Amen.